Great. So our next speaker is Ashley Curran. Ashley, hopefully you can, very nice to see you. Perfect. If you'd like to share your screen, that would be great. And I shall introduce you. So Ashley Curran received her PhD in immunology from Johns Hopkins University School of Medicine and is now a postdoctoral fellow at the Medical University in Vienna. Her doctoral research focused on the molecular mechanisms underlying the pathogenesis of citrullination-directed autoimmunity in rheumatoid arthritis, with a particular focus on the role of MHC class II antigen processing and presentation in the loss of CD4 T cell tolerance. So I'm really looking forward to hearing Ashley's talk. It's a subject that I find really interesting as well. So her title is Citrullination Modulates Antigen Processing and Presentation by Revealing Cryptic Epitopes in RA.
Great, thank you for the introduction and thanks for the invitation. Today I'll be presenting my doctoral dissertation work from Johns Hopkins, which I did in the lab of Dr. Erica Dara, titled Citrullination Modulates Antigen Processing and Presentation by Revealing Cryptic Epitopes in RA, as you said. So again, my focus has been on the pathogenesis of rheumatoid arthritis. The susceptibility to RA is in large part due to genetic risk factors, such as particular HLA-DRB1 alleles called shared epitope alleles. And then in the initiation phase of the autoimmunity, which occurs actually in preclinical RA, so in an asymptomatic phase, there's the accumulation of post-translational modifications, such as citrullination, and then the subsequent formation of autoantibodies against those modifications. So in this case, ACPAs or anti-citrullinated protein antibodies. And these are actually associated, the formation of ACPAs is associated with the possession of these particular HLA-DRB1 alleles, which suggests that antigen processing, these are class II alleles, so class II antigen processing is involved in the pathogenesis of autoimmunity. And so we're interested in how maybe how citrullination could potentially change antigen processing and presentation to CD4 T cells. And so that was like the focus of my dissertation. So briefly, citrullination is the conversion of positively charged arginine residues to neutral citrulline residues by peptidylarginine deaminase enzymes, or PADs. And their substrates, so PAD2 and PAD4 specifically, isoforms are implicated in RA. And their substrates include common RA autoantigens such as fibrinogen, HNRMP, A2B1, and vimentin. And as citrullination increases hydrophobicity through the change from a positive to a neutral charge, we and others have hypothesized that this may destabilize protein folding through sequential citrullination of exposed arginine residues, destabilization of protein interactions, and then following citrullination of previously unexposed, newly exposed arginine residues. And so again, we're focused on MHC class II processing here. And so just a brief refresher, an antigen-presenting cell, like a dendritic cell, takes in antigen from the external space and then processes these antigens in the M2C compartment by so proteases called cathepsins process antigen into short peptides, which are then loaded onto MHC class II molecules before being brought to the surface to be presented to CD4 T cells. And so this is the pathway we were interested in studying to see if we could help understand how citrullination becomes immunogenic and how these autoreactive responses are initiated. So our sort of hypothesis and early model was that as native antigen is converted to citrullinated antigen, this may change the way proteases see and cleave these antigens and change the way the protein folds. And then there's sort of three ways that we imagine this could lead to the activation of autoreactive T cells. So the first would be modification of an existing peptide by citrullination, so essentially presenting a directly citrullinated epitope. And so this, for a while, has been the sort of leading hypothesis in the field in rheumatology that directly citrullinated epitopes may be the direct link between this genetic association with class II alleles and our antibody responses to citrullination. But there's another way that you can generate sort of neo-epitopes, and in this case, cryptic epitopes, which would be the creation of new peptides that were not previously presented from the native antigen. And so this can occur through, again, changes in proteolytic cleavage or in protein folding, basically releasing and allowing the presentation of a completely unique peptide. And finally, going along with this, you can also get the destruction of previously dominant peptides through, again, the creation of cleavage sites potentially inside of this region, or changing and maybe masking these regions through new protein folding and leading to the destruction of that peptide. And so this would result in the presentation of this cryptic peptide repertoire, which could be potentially immunogenic and lead to the activation of autoreactive T cells. And so that was sort of our hypothesis going in. So our first big experiment was what we call ProteMap. So this is proteolytic mapping using a cocktail of MHC class 2 associated cathepsins. So first we wanted to focus just in this M2C compartment to understand how these cathepsins are affected by citrullination, and really understand what this potential total peptide repertoire that is then going to interact with MHC class 2 molecules, how that changes in response to citrullination. So to do this, we used cathepsins B, S, and H. So this is a cocktail that was identified by Dr. Scheherazade Sadiq Nasseri at Johns Hopkins. And she found that these three cathepsins can in vitro recapitulate MHC class 2 processing and generate the dominant peptide repertoire that you would expect to see from a given antigen. So we combine these three cathepsins with our native or citrulinated antigen, allow them to be processed, and then identify the peptide repertoires by mass spectrometry. And so we found that citrullination does alter antigen processing, resulting in the generation of cryptic peptides and the destruction of previously dominant peptides. So here we're looking at vimentin, one of a common autoantigen in RA. This is the amino acid sequence. And the blue here, it represents citrullination sites. And here you're seeing enrichment of regions of the protein in either enrichment in the citrulinated, either PAD2 or PAD4 citrulinated samples compared to native. And so you can see that there's enrichment and depletion of regions in the citrulinated samples along the entire protein sequence. And you can see here on a peptide level, when we look at fold change in these volcano plots, that these citrulinated samples here, you generate these uniquely created peptides. So this is our cryptic peptide repertoire, as we call it. And you see the destruction of previously dominant peptides, as we sort of expected. And when looking at sort of the mechanisms underlying these changes in processing, we wanted to know where these were occurring relative to citrulinated sites. And so we found both proximal and distal changes in response to citrullination. So when we look at created and destroyed sites in these regions in the protein, most of these sites do not actually contain directly a citrulline residue. So this suggests again that there's going to be these distal changes in processing, but this could be either in the cleavage site, but sort of directly outside of the presented region or the newly generated region, or this could be quite distal to the region itself. And so first we looked at newly created cleavage sites in the citrulinated samples and found an enrichment of citrulline residues in the P1 and P2 prime positions. So this again suggests that citrullination is directly changing and altering proteolytic cleavage. But when we look at the created and destroyed regions and model the actual distance to the nearest citrulline versus the expected distance to the nearest citrulline, we find very many regions, particularly in the created category, that are much farther away than you would expect from the nearest citrulline. So again, this is showing us that the citrullination also induces really distal changes in processing, which is likely due to changes in protein folding rather than directly related to proteolytic cleavage. And so to understand how this effect would show up in sort of a cellular model, we used a natural antigen processing assay to sort of see when we do the same thing with native and citrulinated antigens, what is actually presented on the surface of these monocytes, in this case, monocyte-derived dendritic cells, and what would the T cells really be seeing. So we pulse monocyte-derived dendritic cells, as I mentioned, with native or citrulinated antigen, allow them to process and present antigen on their surface. We then lyse the cells, immunoprecipitate the MHC class II molecules, elute peptide, and identify the peptide repertoires, again, by mass spec. So we again see that citrullination alters as well the naturally presented peptide repertoire of human MODCs. So here we're looking at fibrinogen as our antigen, which has 3 chains, alpha, beta, and gamma. And we pulse with either native PAD2 or PAD4 citrulinated fibrinogen. And so we see when we feed native versus citrulinated antigen, first, the creation of cryptic peptides, so peptides that are only presented in this citrulinated condition. So this is sort of the base creation of our cryptic peptide repertoire. Interestingly, we did not find any directly citrulinated peptides, which is sort of in line with findings that for a while in the field that people have been looking in vivo in patients for citrulinated peptides actually being presented, and they have not been found yet. So this suggests, again, maybe that this mechanism, the relationship between citrullination, antigen processing, and autoantibodies may not be directly through a citrulinated neoepitope, but rather just a new cryptic peptide. And we additionally find the destruction of previously dominant peptides, which are only presented in the native and not at all in the citrulinated samples. And interestingly, all 3 of these destroyed peptides contained a citrullination site in the citrulinated samples, which again suggests that the citrullination could then result in enhanced cleavage or altered cleavage, in this case enhanced, and maybe destroy these peptides, which we found really promising. So next, we wanted to sort of investigate the binding of these repertoires to the HLA-DR allele, because the big question has always been sort of how these particular alleles are related to citrullination and why they are associated specifically with the creation of the autoimmune response. And so we looked at using the ProImmune Reveal assay, which can approximate binding affinity to different HLA alleles. We used HLA-DR 0401, which is the most common shared epitope allele that I mentioned before. And so we looked at the sort of native antigen presented repertoire, so all the peptides presented from native fibrinogen versus the repertoire presented from citrulinated fibrinogen. And we find that the binding affinity of the citrulinated repertoire is actually higher than the native antigen repertoire. And when we look at are peptides that contained a citrullination site and model the binding to DR, or measure the binding of to DR 0401 of the native or the versus the citrulinated version of the peptides. We find sort of 3 possible outcomes. So either the enhancement of binding upon citrullination, the ablation of binding, or no change in binding upon citrullination. And so this is sort of in line with existing literature, where some studies have found an enhancement of binding of certain peptides when they're citrulinated to different shared epitope alleles, whereas others find really no change. And so it's been unclear for a while whether that is really the primary mechanism here. But our findings sort of agree that it's not so clear, just as containing a citrulline, in the peptide or not, really leading to this connection between these DR alleles and our T cells. And so we wanted then to look at whether this cryptic peptide repertoire really seems to be immunogenic in RA patients. And so we stimulated ACPA positive RA patients, CD4 T cells. So ACPA positive patients are those that have these anti-citrullinated protein antibodies and anti-citrulline responses, and compared them to patients either without anti-citrulline responses or healthy controls. And so we stimulated them with our created peptide repertoire here. And we did find, looking at activated T cells, we find more activated T cells in the ACPA-positive patients than in the control groups. And here you can see again with stats, the percent of patients responding to the created peptides here are much higher in those that possess these anti-citrulline responses compared to our controls. And then we did a follow-up study with fibrinogen tetramers, also generated actually by ProImmune. So these are tetramers that contain our created peptide repertoire. And again, we find elevated levels of tetramer-specific cells in the ACPA-positive patients compared to the ACPA-negative patient controls. We do actually find a significant number of tetramer-positive healthy controls, but these T cells are primarily turned out to be Tregs. And this is, again, in line with recent studies that have shown that healthy controls and autoimmune patients actually harbor a similar number of autoreactive cells in their periphery, but that most of them are Tregs in the healthy controls. And so these are necessary for controlling the large number of autoreactive T cells that actually do survive T cell education in the thymus. Yeah, and so that kind of leads me to our model, where our results support this model where during T cell education in the thymus, you have the presentation of dominant peptides from given autoantigens, in this case, just self-antigens. The T cells that are autoreactive and bind with too high affinity to dominantly presented peptides will be deleted through negative selection, whereas T cells that may ultimately be reactive to our cryptic peptides will not see their antigen presented in the thymus and will be released as tolerized T cells into the periphery. And then once they're in the periphery, when in this case we have an RA, this citrullinated proteins being released in an inflamed environment, they can then undergo altered antigen processing, be presented. We can then present this cryptic epitopes, these cryptic peptide repertoire on the surface of these cells, and which may lead then to autoreactive T cell activation, which we hypothesize to then provide T cell help to B cells and lead to autoantibody production, in this case ACPAs, which then participate again in this pathogenic sort of feed-forward loop, leading to more inflammation and causing RA pathogenesis. These are my acknowledgments, and yeah, thank you for your attention, and I'm happy to take any questions.
Great, thank you very much indeed, Ashley. That's a fascinating story. Do we have any questions for Ashley? Feel free to put them in the chat. We have time for a couple. I guess not. You must have answered everything very, very clearly, Ashley. So that's great. I put a link to the paper in the chat as well, so people can kind of look there for more details as well. But no, that's fantastic, and we really appreciate you joining us. But I think we'll move on to our next speaker now. But yeah, thank you very much indeed. Appreciate it. Bye bye.